Okay, so this is the end of lesson 5 from the other day. Okay, we finished off at active transport and we didn't get to endocytosis and exocytosis. Right? Now, just so you know, diffusion, osmosis, and active transport are the most important cell transport methods in this course. Okay? What we're going to talk about today is far less important. And what I mean by that is this might be in the multiple choice. The other 3 will all be in the written response of your unit exam. Okay? So you need to know them in far greater detail. Okay? And I'm telling you right now, do not write endocytosis, exocytosis, and explain how they work, or any question in the written response, okay, of your unit exam. I am not asking about these methods in the written. Okay? And the reason I'm not doing that is because they're essentially all the same. Okay? They all involve a mechanical change in shape of the membrane. All right? Active transport, the stuff goes through those little carrier proteins. The membrane itself isn't altered. Okay? Osmosis, the water passes through the membrane. The membrane itself isn't altered. Okay? And large amounts of stuff can move by those methods. But everything that moves by diffusion, osmosis, and active transport is small. Sometimes cells need to either get rid of something large or take in something large. Okay? Something large can't pass through the membrane. Okay? It would be like trying to drive a dump truck through the small gate that leads from the front yard to the backyard. Okay? Like that's designed for a person to go through. Okay? Or a lawnmower or maybe a wheelbarrow, but not a dump truck. Okay? So these involve moving dump trucks, big things, okay, into the cell. They can't pass through the membrane. So what the cell does instead is it changes the shape of the membrane and engulfs the material if it's taking it in. If it's trying to get rid of something big or really large amounts of something quickly, okay, then a vesicle will move from the Golgi apparatus to the membrane. Remember that vesicle is made of the same materials as the membrane. So once it gets to the membrane, it just merges with it, okay, and just kind of pops open, and whatever was inside is now out. Okay? For taking stuff in, the process is a lot like this. I don't know what's grossed out by this. But if you have a dog and you take them for a walk, you have to clean up after them. Okay? And you get those little bags that go around your hand, okay? Little poop bags. Okay, and what you do is put the poop bag on first. That is like the most important step. Okay, put the poop bag on first.
Just like hold it open and like.
No, don't do that. Put it over your hand like a glove. Okay, and then you just you pick up the poop and then you turn the bag inside out. Okay, now everything is inside the bag and you haven't had to touch it. That's exactly the same process that a cell uses to engulf something. Okay, that thing just touches the membrane, okay? The membrane just goes around it and then basically turns inside out and the thing is inside now. Okay? So, for exocytosis, remember we said that these are the 2 functions of the cell membrane, right? Endocytosis and exocytosis. So exocytosis is the getting rid of stuff. Okay? It's either something the cell has produced that it wants to export, like hormones or something like that, or it's waste. Either way, the process is the same. The vesicle, the transport vesicle, goes to the membrane, merges with the membrane, and pops. Okay? Whatever's inside it is now out. Okay? There's actually 3 kinds of endocytosis. But again, the mechanics is the same. All that's different is what's coming in. Okay? So, if a cell is like really dehydrated,
Sorry for the interruption, but there's a vehicle in the student parking lot, a RAV4 license CR.
All right, so if a cell gets really dehydrated, it will have to take in a lot of water. And osmosis is only going to move so much water. It's a fairly slow process. Okay? So what a cell can do is it can gulp. Okay? And it will just start gulping in huge amounts of the fluid that surrounds it, because all cells are surrounded by what's called interstitial or intracellular fluid. Okay? And so it will just gulp it. The membrane will just fold in and engulf a whole bunch of water, rapidly bring it into the cell. That is called pinocytosis. Okay? if a cell needs to bring in a large solid particle that's too big to pass through the membrane, it will engulf it through a process called phagocytosis. Okay? That's essentially cell eating. So pinocytosis is cell drinking, phagocytosis is cell eating. Okay? And then there is the final one, which is receptor-mediated endocytosis, or RME. Okay? It's, again, mechanically identical. The cell membrane just folds in on itself, but it's triggered by receptor molecules that are embedded in the membrane. Remember we talked about how the membrane is like Jell-O salad? Okay? So there's receptor molecules all over the membrane. Okay? When a chemical signal touches one of those receptors, it signals the membrane to engulf it and take it into the cell rapidly.
What is intercellular fluid made of? Is it just
Mostly water. Yeah, I mean, there'd be some other solutes dissolved in it, but mostly water. So these receptors, like I said, they get triggered by the signal being, or touching them or being accepted, and then the cell membrane just envelops them and brings it into the cell so that it can affect whatever it's supposed to do. Okay, so you'd have receptors on your cell membranes for adrenaline. You would have receptors on your cell membrane for insulin, for estrogen, for testosterone. All of the hormones all have their own receptors all over the cell membrane. Okay, there's literally an antenna for every signal that the cell can receive. Okay, it doesn't work like the radio in your car where one antenna receives all the signals. There's actually a signal for, an antenna for every signal. Okay, that's why the cell membrane is so much more complex than most diagrams make it look like. All right, so phagocytosis, pinocytosis, receptor-mediated endocytosis. They're all the same except for what they take in: water, food, hormones. Okay, multiple choice questions only. As long as you can recognize it, you're good with those. All right, so if we're looking at phagocytosis, one of the main cells in your body that does that are your white blood cells. Okay, they will engulf bacteria and other harmful things in your body and basically use them for food. A lysosome will merge with a vacuole that contains a bacterium, okay, and it will eat it. Okay. For pinocytosis, okay, that's taking in the interstitial fluid, okay, or intercellular fluid, and this is actually an electron microscope picture of what that looks like. Okay, so each one of these, okay, is one of those pinocytic vesicles being formed.
That's weird.
Yeah, it's really funky. Okay, so they probably just like artificially somehow dehydrated a cell and then just put it back in water and put an electron microscope over it to get a shot at what it looked like.
So then those like little bubbles just like pop inside of the cell?
Or they'll go to wherever they're needed. Yeah, and then of course they'll merge, because all membranes are made of the same material, right? So they'll merge with it and bring the water along. Now, in terms of microscopy, what's the disadvantage? Like an electron microscope shows us things greatly magnified, right? Like it shows us things we can't possibly see with a light microscope. What's the disadvantage?
No color?
No color. They're usually artificially colored by computer. Okay. What else? Depends on the type. You can on this one. This is a transmission electron microscope, so there's no depth. You're right. It's two-dimensional. A scanning electron microscope. You've probably seen like they do pictures of like bugs, and you can see every little detail of a bug on a scanning electron microscope. They actually have to coat the specimen with a fine coating of gold, and then do the electron microscope picture of it. It's very expensive, but also a very cool process. But the disadvantage here is it's dead. Okay. Once you turn on the electrons, you're essentially bathing it in radiation, okay, and it's dead after that. Also, for a lot of those, the prep process, like the coating in gold, okay, is destructive. Okay, it's going to kill whatever it was you were observing. So the advantage of a light microscope is you can observe a process happening. A scanning electron or transmission electron microscope, you can take a picture of it, and that's it. You can't see anymore. Okay, you get a freeze frame of one instant.
Oh wait, so all those bugs are dead?
Yeah,
Wow, that's kind of sad.
that's good bugs.
But they're dead.
Then they're good bugs. Okay, and then lastly, we had the receptor-mediated endocytosis. So we said you get a receptor that is triggered by a hormone or something like that, okay, and then it is taken in, and this is what that looks like under an electron microscope. So you can actually see here, this is the RME vesicle that's being formed, and you can actually see here is a receptor, and there is the molecule sitting on it, and there's actually many of those inside that vesicle. Okay, so that diagram there isn't all that far off, okay, from what it actually looks like. It's pretty crazy, yeah. Okay, all right. I would like you guys to see if you can answer these questions. You're not necessarily going to find the answer for every one of these in that lesson. I want you to think about it a little bit, okay? And then we'll talk about the answers here. Okay, so for question number 1, color blindness is caused by a person's cells being unable to produce certain proteins. Where in the cell does the problem lie?
Like the ribosomes or the nucleus or nucleolus, because it's not polymerase DNA, or ribosomes because it's not producing it well.
Right, it's actually not the ribosomes. The ribosomes are working fine if they're only unable to produce certain proteins. The problem is in the nucleus. The problem is with your DNA. Color blindness is a genetic mutation. It's hereditary. It's actually also X-linked, which means males are far more likely to have it than females. Okay? It is caused simply by your cells having the inability to produce proteins that are necessary for the cones in your eyes to produce certain proteins that allow them to distinguish different wavelengths of light. Okay? Now, color blindness comes in a huge spectrum, oddly enough. It's color blindness. Okay? But a huge spectrum of like degrees. Right? Some people have like red-green color blindness, which means like on your driver's test is probably when you find out you have that. Right? They show you the little dots, and you can't see the number pattern inside the dots because you can't tell the difference between the two colors. Okay? There's many different kinds of color blindness. Mr. Van Erp, who's supping across the hall right now, used to be the biology teacher here. He's completely colorblind. And I mean completely. Dude sees black and white. Okay? And for his retirement gift, we got him those glasses that help colorblind people see colors. It was like somebody slapped him in the face when he put them on. Okay? It's like, that's green? Oh, like he'd never seen, they just didn't know. And we always used to kind of make fun of him because you could tell if he got out of the house before his wife saw him. Because he'd come in wearing like the most ridiculous color combinations. And one day he came in and he was wearing this orange and blue shirt with like light gray pants. And I was wearing the same pattern of shirt with blue pants, light blue pants. But it was exactly the same pattern, but mine was red and blue. Right? And he's like, hey, we're twins. Yeah! And I walked down the hall, and on my arm, yeah, look at us, we're twins. Look, he had no idea we were twins. I was kind of mean in retrospect, but it was kind of funny. Everyone got that he was colorblind and just couldn't tell the difference. Like, hey, Jerry, is that red? Yeah. Okay, sure it is, bud. Yeah. But yeah, it's totally genetic. He's got exceptional color blindness, and he can't see any of them. This is actually why a lot of traffic things are done the way they're done. Okay, on a traffic light, where's the red light all the time? Top or far left. If it's horizontal, it's far left. Okay, if it's vertical, it's always on the top. Okay, green is always on the bottom, yellow is always in the middle. Okay, if you have an advanced green, like the little arrow, it flashes. But when it goes to yellow, it also goes solid. These are all for people who are colorblind. Okay, Mr. Van Erp would have no idea whether that light was yellow or green. But because it's flashing when it's green, he knows it's green. He also knows it's green because it's on the bottom, not on the top, because he can't tell the difference between the red one and the green one. Okay? He's that colorblind, but that's why we set things like that up. Okay? It's because he totally can't see the difference. It's also why there's such a major contrast on all road signs. Yellow background, black text. Okay? Red background, white text. It has to be that much of a difference for a colorblind person to be able to read it. Okay? If you put stop in green on a red background, the nerve would drive right through that. Okay? You wouldn't be able to read it. It would just be an octagon. Okay? Like, ooh, sweet. Wonder what that sign's for. Okay? Because he's colorblind, but it's all genetic. Okay? So the problem is actually in the person's DNA. And now once again, he gets to make fun of me even though he's retired. All right, number 2, why is it important for organelles to have their own membranes? A big long word starts with C. Compartmentalization. Okay, compartmentalization means they all get an ideal environment to carry out the process they carry out. All right, what might happen if the lysosome's membrane ruptured?
It's gonna eat the cell.
It'll eat the cell, yeah.
Self-destruct.
The rest of the cell, yeah, will self-destruct or be digested. Okay, it would certainly be bad. All right, and what problems might a cell have if all the ER was removed?
Like disintegrate because the ER makes the membranes. So
Yeah.
it's kinda-
My first thought would be a lot of things will go wrong because the ER does a lot of things. It repairs membranes, it detoxifies poisons, it breaks down complex carbohydrates, transports protein if we're doing the rough ER, okay, and can produce fat-soluble hormones. Well, it's a lot of things. If that's suddenly gone, that cell is in real trouble. It's like cutting off all our arms and legs. Okay, like it would be very difficult to do anything. Okay, after that. All right. Questions on any of those? Okay. Okay, so we're going to look at the multicellularity stuff here, guys. We probably won't get all the way through it, but that's okay. We can finish it up on Friday, but we'll get it started at least. Okay, so we're kind of done looking at cells individually. Now we're going to look at still similar processes because your multicellular organism is still going to use the same processes, but on a larger scale. Okay, so multicellularity and specialization. Now that is a very, very important term. Okay. If you are truly multicellular, you have specialization. Okay. It is possible to have an organism that is made up of more than one cell, but is not multicellular. Okay? To be truly multicellular, your cells have to be specialized to do different jobs. Okay? We definitely have that. Okay? We are a true multicellular organism because your muscle cells are different from the cells of your small intestine, from the cells in your heart, from the cells in your bones, from the cells in your brain. Okay? They're all very, very different. At their core, they're all cells. They have a nucleus, they have ER, all of that kind of stuff. Okay? But they're specialized to do a certain job, which means their structure is different. And maybe the amounts of organelles of a certain kind is different. Okay? As an example, muscle cells use huge amounts of energy, okay? Exert fairly large forces, and are subjected to a lot of stretching and things like that. So as a result, muscle cells have way more cytoskeleton, that would be the microfibers and microfilaments, running through them to maintain their shape. Okay? If you've ever heard this, like when you work out, you're breaking muscle fibers and then they heal back, that's, you're breaking cells, you're breaking, and they heal back stronger, okay, afterwards. Okay? They also have lots of mitochondria because they have to carry out a lot of cellular respiration. They're an energy demanding cell. Okay? They would have way more mitochondria than a cell in your stomach. Okay? A cell in your stomach basically just has to make enzymes and stomach acid. Okay? It doesn't have a lot of other jobs to do. Okay? So it doesn't require nearly as much energy as a muscle cell would. Okay? Does everyone get the idea of what specialization means? Okay? The cells at the basic level are similar, but they are specialized to do a certain job. Okay? Specialization is that key characteristic, okay, or what's the word I'm looking for? I'm drawing a blank here. Oh, I hate it when that happens. characteristic, we'll say, okay, of property, that's the word I was looking for, okay, of a multicellular organism. Got to have cells that are specialized. Okay, so we're going to look at how multicellularity evolved. Okay, we're going to look at the role of specialization in multicellularity. In other words, you have to have it or you're not. Okay, and examine the plant as an example of a multicellular organism with specialized cells and tissues. We actually don't look at animals in Science 10. Okay, in Science 10, the plant is our multicellular organism example. Right? In biology 20 and biology 30, you do a lot more with animals. You look at different organ systems each year. So like, I think you do digestion in bio 20 and circulatory. And then in bio 30, you do excretory and nervous and muscular or something like that. Okay? So you do more animals in bio, okay, not in Science 10. Okay, so multicellularity has actually evolved separately, okay? Unlike, the similarities between the cells, that evolved from a common ancestor. Multicellularity actually evolved separately in several different groups of organisms, okay? So it has evolved in plants, it has evolved in animals, and it has evolved in fungi. Okay? There are also, evidently, a few extinct groups, okay, that may have also been multicellular, okay, that are a little bit different or offshoots of those. Okay? But it actually did evolve separately, okay, in 3 different groups. All right. Now, looking at, okay, our kind of history of the Earth here. Okay, in terms of how long does it take for everything to happen. Okay? Life on Earth is actually, geologically speaking, especially multicellular life, a fairly new thing. Okay? Because the Earth is 4.5 billion, 4.6 billion years old. Right? But it spent a lot of its life being a molten ball of rock, okay, which was completely inhospitable to life. Okay, so if we go back, okay, not so much to the beginning of the universe with the Big Bang and all of that. Okay, if we just kind of go back to the start of our solar system, okay, and you have your protoplanetary disk and the formation of the sun. That's about 5 billion years ago or so, when the sun kind of finally ignited and started carrying out nuclear fusion and all that. Okay, there would have been a lot of debris around the sun, which then coalesced into the planets. Okay, now that results from things hitting each other. Okay, so 2 big asteroids hit each other, and there's a lot of heat generated in that. They actually merge together, okay, to become one thing. Right? That happens over the course of billions of years, and that forms the planets of our solar system, Earth included. Right? As Earth is just starting to cool, something really bad happens to it. Anyone know what it is?
Another planet crashes into it.
Another planet crashes into it. Just as Earth is starting to get things together, okay, somebody comes along and smashes into it. About a Mars-sized object called Theia. Okay? This is actually what formed the moon. This impact actually caused the moon to form. Earth did not have a moon before that time. Okay? So Theia comes in and hits the Earth, and it's not like head-on like this. It's kind of a glancing blow. Right? So it catches it just on the edge. And all that essentially does is, annihilates both of them, really. Okay? There's, the larger mass of the Earth kind of stays together, but it becomes molten again, and a lot of the core of the impactor is absorbed into the Earth. Okay? So all the heavy parts, okay, are absorbed into the Earth. Meanwhile, the crust of the Earth and the crust of this impactor kind of get thrown out into space from the impact. Okay? So now there's this hot molten ball of really heavy stuff in the middle, and there's this cloud of debris around the planet after that. Well, that cloud of debris eventually coalesces and forms the moon. Okay? So now we have 2 molten balls orbiting each other. Okay? That would be the early Earth and early moon. It takes a long time for Earth to cool for a lot of reasons. That made a lot of heat being smashed into. It makes things really hot. Okay? But the other part of it is Earth and the moon are really, really, really close together. Okay? And the gravity between Earth and the moon keeps pulling on each other. Okay? And it's kind of like if you take clay in your hand and you start, molding it around, it gets warm. Okay, the particles in the clay rub against each other as you knead the clay. Okay, well, the same thing happens with gravity. Okay, gravity of one thing pulls on the other, and it twists it and compacts it and pulls on it, and it makes it hotter. Okay, that interaction is still going on between Earth and the Moon. Okay, it causes what? Tides. Yeah, we still feel the effect of the Moon's gravity because it pulls all the liquid on Earth towards it. Okay, and that results in the high tides and low tides as the Earth rotates underneath the Moon. All right, so that stuff is all going on. Moon is very close to the Earth. Earth starts to cool, okay? But a lot of changes have to happen on the Earth before anything even remotely resembling life can start forming, okay? So it's going to be, what do we got here? This is like 4 point something billion years ago here, okay? We start getting like DNA and prokaryotic cells kind of right in here about 3.9 billion years ago or so. Okay, we start getting 3.7 billion years ago a cooling ocean. Okay, that's forming. This stuff here all happened kind of due to ponds evaporating. Okay, and the stuff that got left behind kind of formed first proteins and organic materials and stuff like that. Okay, and then you get the first photosynthetic cells. Okay, and once you start getting life, life starts to change the whole planet. Okay. Early Earth, when those first photosynthetic cells appeared, had a very different atmosphere. It would have been incredibly toxic to us. Okay. Would have had ammonia, lots of carbon dioxide, okay, lots of horrible stuff that we couldn't breathe. Okay. But the little algae were perfectly happy with that because it had lots and lots of carbon dioxide and they were in the water. Okay. There's no life on land for like another 3 billion years or so. Okay. So you start getting this environment where photosynthesis takes place. Now, if you're a plant and there are no predators, can you go crazy? Absolutely, right? There's nothing to stop you except possibly running out of your raw materials, of which there are, well, what would seem like an endless supply. Okay? So photosynthesis goes on and it just takes off. Okay? But it starts to radically change the composition of the atmosphere. We know carbon dioxide is a greenhouse gas, right? So it made the Earth much warmer. But as photosynthesis started to take it out of the atmosphere, it started to put a lot of oxygen into the atmosphere, which is not a greenhouse gas. And it plunged the Earth into an ice age. Okay? I don't know if anyone's heard of this period of time called snowball Earth. Okay? It's actually a period of time where Earth was actually literally a giant ball of ice. Okay? Because the Earth's temperature cooled because of all the removal of carbon dioxide. Well, once it froze, there were not nearly as many photosynthetic organisms and things started to go back the other way and we started getting more of a balance. Okay? That was actually the first mass extinction of life on Earth. Okay? Earth has faced many of those in its history where some condition changed very quickly and everything just dies off really fast. Okay? So we've got your global ocean going on there. Okay? You got lots of kind of other organisms forming here. Okay, everything up until this point here, okay, is really kind of single celled. Okay, all single celled and all prokaryotic. Okay, you got snowball Earth here. Okay, that's the Huronian Ice Age. The Great Oxygenation, okay, is what causes that. Okay, the earliest multicellular organisms are present 2.1 billion years ago. So less than half of Earth's history contained multicellular organisms. And we're talking very simple ones at this point. But once you get multicellular organisms, things take off. Because now there's all these niches that need to be filled. Okay? And multicellular organisms have the ability to specialize and fill those niches very quickly. Okay? So we start looking here. Everything is also still on Earth, or still on Earth, in the oceans. Okay? There's nothing on land yet. Okay? We don't get to having multicellular organisms on land until right about here, okay, about 500 million years ago. Okay, or so. That's when life finally started to come out on land. The fish with the big flippers, okay, they could go out on land for a little while and then come back, and that led to amphibians and reptiles and so on. Okay, so it's a very long period of time before multicellularity evolves. Okay, but when it does, it's a huge advantage. Okay, why is being multicellular such a huge advantage over being a single-celled organism? Adaptability, yes. And something that we talked about yesterday,
You can get bigger.
you can get bigger. Okay? Yep, the fossil record has shown us that on most occasions, bigger has been better. Okay? There have obviously been isolated times throughout the Earth's history where it was not. Okay? Like being a dinosaur 65 million years ago, being bigger was not better. Okay? But in most cases, being bigger has been better. Okay? It makes you able to access more resources, and fewer things want to eat you. Okay? Because you're big and hard to eat. So multicellularity, okay, allowed more complexity, greater survivability, greater adaptability, okay, all those kinds of things. So it was a huge deal when it appeared in the fossil record, and then things just kind of take off. Okay, so what distinguishes a multicellular organism from a colony of cells is division of labor, which is another way of saying what word? Specialization, yeah. Okay, so is the organism you see there a multicellular organism? It's got more than one cell, but is it truly multicellular?
No.
It's not. It's a colony of cooperating cells. All the cells are identical. There's no specialization there. They all feed, they all produce waste, okay, they do all of that stuff. But no one of them is better at a single job than the others. They're all identical. That's a colony, not a multicellular organism. Okay. So that's the big kind of stepping stone or dividing line between a colony of cells and a multicellular organism. Specialization is that key characteristic. Okay. So the evolution involved increasing specialization and division of labor as multicellular organisms became more and more complex. Okay, and colonial aggregates, like the thing you saw on the last slide, all cells may have been motile, that means they could move, okay, with flagella, okay, as cells in the colony tend to become increasingly dependent on each other, okay, some of them may have lost their ability to do one thing, and others gained a greater ability to do it, and eventually over time that can lead to specialization and a true multicellular organism. Okay, now, in these pictures here, early Earth, what do you notice in all of these pictures?
Looks kind of nasty.
It looks nasty, okay? There's a lot of like volcanic activity. And what did you say about the moon?
I said smoke.
Oh, smoke.
And it was big.
Moon's big in all of the, well, not that, there's no moon in the third picture, but okay, the other two, the moon is really, really big. Why?
Because it slowly drifts away and hasn't drifted away yet.
Yeah, exactly. The moon is not bigger, it's closer. Okay? When the moon first formed, it was incredibly close to the Earth. It has been steadily drifting away at about 2 centimeters per year since its formation. Okay? So this has a lot of effects because the closer it is, the stronger it can pull on the Earth. So the more tectonically active Earth will be, okay, because of all this gravitational kneading of the clay. Okay? But the other thing is, it also affects the Earth's rotational period, how quickly it rotates on its axis, so the length of a day, okay? And tides. Okay? The Earth had a liquid ocean when the Earth was a lot closer to it. Can you imagine what tides would be like if the moon was 10 times closer to the Earth than it is right now? Okay? Like, it would be like if you had a container full of water and you just sloshed it back and forth. Okay? That would be what it would be like as the Earth rotates underneath the moon. Now, remember, not only is the moon closer, making the tides greater, but the Earth is rotating faster. Okay? The Earth's day has been as short as about 6.5 hours. Okay? When the moon was closer, Earth spun a lot faster. As the moon goes away, Earth gets slower and slower. The moon has already stopped rotating. It's tidally locked. We only ever see one side of it. Okay? Eventually, as it goes further away, okay, the Earth will stop rotating on its axis and become tidally locked to the sun. Okay? As is the case with like Mercury and Venus. Okay? They're essentially tidally locked. Okay? So it's much closer, and because of that, it's going to pull the oceans a lot further. But because the Earth is spinning really, really fast under it, the tides are really close together. Okay? You get like 2 high tides and 2 low tides a day right now. Okay? Imagine if the Earth is spinning 4 times faster. Okay? And it gets 16 a day, right? 16 changes in the tide a day. Imagine being at the Bay of Fundy when that's going on. Okay? It would be pretty crazy. All right, making some sense? So this is actually creating, like, the conditions to us sound terrible, right? Imagine you wouldn't want to live on the coast, okay, if that was the case. But if you're just starting to develop and evolve life, that is a big deal, right? Because the constant sloshing of the ocean is meaning there's always new nutrients, food supply, oxygenation. The more you agitate water, the more oxygen gets into it from the air. Okay, all of those things are good conditions for life. Okay, so even though it doesn't sound like it would have been desirable, it was perfect to get life going on Earth when it happened. Okay, all right, we got time to talk about a few of the plant cells here. So we're going to skip the parenchyma cells though, and the water conducting cells, and food conducting cells. So we're going to talk about those later. Okay, this is something we're going to look at tomorrow. This is the layers of the leaf. Okay, so someone took a leaf, okay, and they cut it this way, and they're looking at it on edge. Right? And what you see in a plant, in a leaf, okay, is that the cells in the leaf all look different. Okay, the cells on the top are kind of round. Okay, the cells underneath are very rectangular, and then you've got some kind of randomly shaped cells underneath that, and then you've got another layer of the kind of rounded cells on the bottom. Okay, why do they all look different?
Different jobs.
They have different jobs. The layers of the leaf, guys, is our example in Science 10 of specialization in a multicellular organism. So mark it in your notes. Being able to explain the layers of the leaf as an example of specialization is something you're going to be asked to do. Okay, we're just introducing it today, but we're going to talk about it lots. Okay? So when you look at the leaf tomorrow, you will see this. Okay? It'll look just like this tomorrow. Okay? Now, each one of those layers has a different job. Okay? The top and bottom layers are called the epidermis, which is another name for skin. Yeah, or outer covering. Epidermis is outer layer. Okay, so skin. Yeah. Okay. Essentially, this is like the skin of the plant. Its job is to secrete a waxy material that coats and seals the leaf. What does that prevent?
Water from entering.
Water from entering, and more importantly, water from leaving. Okay? Plants that have a waxy cuticle, that's what it's called, the waxy material is called the cuticle, okay, can survive periods of drought because water can only evaporate through little holes called stomates, which is another example of specialized cells we'll talk about. Okay? And those are mostly on the bottom of the leaf. Okay? So the upper epidermis doesn't have any stomates. The lower epidermis does. Why would you want the holes through which water evaporates to be on the bottom?
Because the sun that hits the top, it will evaporate faster.
Exactly. They're in the shade. Okay? Less evaporation. Okay? So plants have evolved all these little kind of characteristics in order to enhance their survivability. So you got the upper epidermis, lower epidermis, they secrete this waxy cuticle, which you can feel if you touch a leaf. Okay? And have you ever seen like what happens to a water droplet or a raindrop when it hits the leaf, right? It just beads and then runs off. It never gets absorbed. Leaves do not absorb water. Okay? It runs off the leaf and onto the ground, where the roots can do it. It's a perfect example of something evolving and doing two jobs. Okay? That waxy cuticle ensures that it doesn't get absorbed through the leaf, water doesn't leave through the leaf, but the water ends up at the part of the plant that can actually do something with it. Okay? All right, so epidermis secretes the waxy cuticle. That means that those cells, as you can see, they're not very dark. They're almost transparent. Why is that important? So light can get through, because look at the cells underneath. Are they darker?
Yep.
Are they packed really close together?
Yes.
Yes. Okay, this is what's called the palisade layer. These cells are packed super close together and have an incredibly large number of chloroplasts inside. Because their job is to, right, carry out photosynthesis. This is the photosynthetic layer of the leaf. Not that the layer underneath doesn't carry out some photosynthesis, but it's not its primary job. The primary job of those palisade cells is to be packed in as close as possible and expose as many chloroplasts to the sun as possible. Okay, that's why they look different. Okay? They have a different job. They're specialized to carry out photosynthesis. Okay? underneath the palisade layer, and sometimes you'll see 2 layers of palisade cells, that's not abnormal. Okay? Underneath that, you see that the cells are no longer like rectangular, and they're no longer tightly packed together. They're kind of irregular, and there's gaps in between them. Okay? This is called the spongy layer, okay? Because it is kind of like a sponge. Water fills those gaps between the cells, and this is where the chemical parts of photosynthesis can happen. Okay? In photosynthesis, carbon dioxide needs to come in, oxygen needs to go out, and water has to evaporate, okay, in order for all of that to occur. So all of those things are held in the gaps between the cells in the spongy layer. So they're specialized to have an irregular shape that creates space for that chemical exchange to happen. Okay, so are we seeing the idea here of specialization? Everything is a little bit different, right? And if you look at these cells, they generally also don't look like they're terribly dark. Okay? They don't have as many chloroplasts in them. Okay? Their cell walls are far more irregular. Okay? Their shape is far more irregular, okay? Because they have a different job. And then on the bottom, okay, you've got that epidermal layer again, but you can actually see in a couple of places here that there are gaps. Okay? Those are the stomates. Okay? A stomate is made of 2 cells. The cells are called guard cells. They're also specialized. Okay? This is how they're specialized. Their water vacuoles can fill and empty essentially on command. Okay? When a plant starts to get stressed for water, the guard cells will empty their vacuoles. It happens because of osmosis. Salt just starts to build up around the guard cells, and water is naturally drawn out of them by osmosis. When the water vacuoles get emptied, the guard cells change shape and they become flat and closes off the stomate. Then overnight, as the plant recovers its water supply and it's cool, okay, all of those water vacuoles fill back up. And when they fill back up, they become bean-shaped and the stomate opens. Okay? It's all controlled by osmosis. The plant doesn't decide, oh, I'm going to close the stomates now. Okay? It just happens naturally by osmosis. Okay? But those cells are specialized to be able to change their shape in response to the amount of water that's present and control evaporation. Okay? So here's what's going to be expected of you. I'm going to put a picture of a leaf on your exam, like these. Okay? And I'm going to ask you to tell me why it's an example of a multicellular organism, and you're going to talk about specialization, and you're going to tell me some examples. The epidermis does this. Okay? The palisade layer cells are specialized to do this because they have this in them. Okay? The guard cells, they can do this. They're specialized to do this job. You need to be able to tell me, here's an example of a multicellular organism. Here's why it's multicellular, and here's some of the examples of specialization within it. Okay? That sort of makes sense? That's kind of the end result that we're looking for, okay, in terms of that understanding. Okay, this is a little bit better diagram of it, but okay, you can see here, this is an electron microscope picture of the layers of the leaf. So here is your palisade layer, so you can see all those individual cells packed right in there. Okay, these are your epidermal cells on top. Okay, right here, that's a vein. If you've ever looked at a leaf, you can see the veins running through it. Okay, and the veins have 2 different kinds of tubes in them. Okay, these tubes here in the middle transport water. These tubes on the outside transport sugar.
So is that thing covered in gold? Is that looks like it?
Yes, that would be a scanning electron micrograph. Yeah. Okay, and so this is what the artist's impression of that would look like. Okay, you got the spongy layer, you got the stomates and guard cells underneath. Don't know if that showed up in the electron microscope picture.
You get a kind of notification.
Yeah, you need to upload that.
Did I touch that? Here, wait. Where's my arrow?
It's the right,
Middle of the
to the right,
screen.
to the right.
This?
Yes.
No, that's not it. That's for my smartboard. That's different. There we go. Okay. So actually, here's the bottom layer. So you can kind of see right here is probably a stomate. This would be one guard cell. That would be the other. That stomate would be open. Okay, so that's the kind of stuff we're going to see tomorrow. Okay, like you're probably looking at this and going, I'm never going to see something like that. You'd be surprised just how much what you look at under the microscope is going to look just like this. Okay? All right. Hello.